So, welcome to everybody. Nice to meet you. I'm Matteo. It's a great pleasure to present Marco, Professor Marco Gerdol. We were at the same university in Trieste, University of Trieste. We did functional genomics together, and then we went for, let's say, different PhD path. I'm more in the structural biology field. He did environmental biology in 2012. Then he became a researcher and then tenure track researcher at the University of Trieste. And then now, since this year in February, right?
Last year, yeah.
Last year in February, he became associate professor in genomics. It's a pleasure not only for this, to present Marco, but also because he is a real expert in genetics and genomic sciences. The main expertise involves mainly the study of the genome, the study of how the genome changes in our cells, how this can drive adaptation for example, specific organism, how this can drive the response to tumor, the response to immune system, and so on. And we will see something today in this talk. To give you a few numbers, he is a real expert, and considering also the relatively young age, 40. He has more than 130 papers, scientific papers, an H index of 31, and more than 3,500 citations, which is quite a lot, considering also the biology field. He is also editor of BMC Genomics, and today he will present this talk beyond single nucleotide polymorphism, how are large-scale genome sequences approaches revolutionizing our understanding of inter-individual diversity. Before you can start, I want to say we will have some minutes, let's say a few minutes, 10, 15 minutes for questions or curiosity. You can ask whatever you want, and then we have a short break downstairs with a bit of wine and foods and so on. So in case we can proceed also downstairs. Marco, please.
Okay, thank you very much for the nice introduction. So I know you have different backgrounds, so I will try to keep things as simple as possible, but hopefully I will provide some interesting news for you, because you're mostly used to think about the genome in terms of the human genome and what comes with its variation. I need to point that. Okay, so just to give you a few numbers about the human genomes, let's remind how did we generate this important resource, which is considered to be one of the hallmark achievements of scientific progress. It took 13 years to obtain the reference genome, which is still the one we are using today. The project started in 1990. It was completed officially in 2013, even though it took only 13 years to obtain the first reference genome. Then there were other 10 years used into its refinement, basically. The cost was extraordinarily high, nearly $3 billion, even though nobody really knows for sure the exact amount of money which was used to obtain this very, very important resource. The goals were obtained in a very long time with the collaboration of multiple laboratories all over the world. And besides the achievement of the human genome sequence, I would say that the most important goals that were met by this project were really putting together different fields of expertise, starting from genomics, but of course also involving people working on the development of the machinery used to obtain the sequences themselves. So people from all over the fields of physics, engineering, and so on were put together. And this also led to an incredible push for the development of the national sequencing centers that nowadays everybody uses in pretty much all countries to obtain sequencing data, not just from human, but for pretty much any living organism. So besides the sequence itself, the Human Genome Project also led to the mapping of nearly 4 million polymorphisms that are extremely important because mutations are what make us different from each other. And they can be linked to phenotypic effects, of course, in terms of difference in age, weight, colors, and so on. But also, of course, they can be associated with disease. And this is a key topic for biomedical research. Besides that, let's keep in mind that most of scientific research is not performed directly on human for obvious reasons, but it uses model organisms, such as mouse or rats, but also model invertebrates, such as Drosophila or Cenoradictis elegans, which is a model worm, for example, that is used for developmental studies. So among all these achievements, we might want to ask ourselves whether the human genome is something really special or not. I'm still having troubles at changing slides, apparently. Okay, I made it. So what's so special about the human genome? Well, first we should have a look at the numbers, just to give you an idea about how large and complex this entity is. You're probably all familiar with the chromosome representation. So our genomic information is split between 22 autosomes plus 2 sexual chromosomes, X and Y. Overall, the sequence is about 3 billion base pairs, 3 billion nucleotides. So we call those 3 gigabases, all right? And these nucleotides are associated with nearly 20,000 protein-coding genes. They may seem quite a lot, but actually researchers that were originally involved in the Human Genome Project thought that the human genome was more likely to harbor something around 1 to 200,000 genes. So they largely overestimated the actual amount of protein-coding genes initially. And the reason why they did that is because they weren't that much aware about the impact of alternative splicing, which generates quite a lot of different mRNA molecules, which nowadays are estimated to be around 400,000, okay? Besides that, we also have other layers of complexity that are not directly provided by variations in gene content, but rather on variation at the epigenetic level. You've probably heard about epigenomics or epigenetics. Well, this is the study of modifications of nucleotides that do not really involve a change into one of the 4 classical nucleotides, A, T, G, or C, but to their methylation status. This regulates the accessibility of chromosomes, chromatin, to transcription factors and has an effect on the way the genes are regulated, basically. And then there is another thing that is extremely important, which is the main topic of today's talk, which is the inter-individual variability, which is provided by mutations, okay? Mutations that can be point mutations or can be more substantial, as I will hopefully explain you. So getting back to our original question, what's so special about our genome compared to other organisms? Well, the answer is pretty simple, but maybe not the one you expect, because the human genome doesn't have anything that special when you compare it to other organisms. As you can see from this graph here, its size is pretty standard, average, I would say. It's not any different from that of any other mammal. It's even much smaller than the genomes of a few other organisms that you may think are relatively simple or primitive. But for example, to use a very popular species here in Slovenia, let's take the olm, all right, that you all know as a symbol of Postojna caves, for example. Well, its genome size is extremely large. It's closer to this end here. So it's a genome that's about 10 times larger than the human one. Even though this may seem a relatively more simple organisms, its genome is not simple at all. And the same thing could be said about several plant genomes that are extremely large. So there is this paradox, which is the lack of correlation between genome size and the apparent morphological complexity of a species, which is called the so-called C-value paradox. But more in general, I would say that we have suffered for many years about our anthropocentric view of evolution, thinking that humans are like the pinnacle of this evolutionary process, the perfect organism, the only one which was able to adapt better than everybody else to planet Earth. This is really not true. We are a relatively poorly adapted species that is highly susceptible to change, and maybe in a few 100,000 years we will completely disappear, and many other species will survive happily even in the world that will come. So, after the Human Genome Project obtained a sequence, a full sequence of nearly, as I told you, 3 billion nucleotides. Well, the question was what to do with this data, what's next? And the opportunities were enormous, obviously. And here I'm reporting one of the views that one of the main scientists involved into the project, Francis Collins, had back in 2003, when the sequence was finally released to the public. Well, it was pretty much imagining a world, a future world, where the ability to obtain genomic sequence data would have been accessible to everybody. So this would have obviously required to decrease the cost of sequencing DNA by several orders of magnitude in order to obtain the genomic sequence, the full complete genomic sequence of an individual for as low as $1,000 or something along these lines, right? And keep in mind also the timing that was required, as I told you. The starting point was 13 years to get the complete sequence and nearly $3 billion, okay? So it's a huge leap to get from these numbers to $1,000 and a few days of work. So, was Francis Collins a visionary or just a fool? Well, it turned out that in the long run, he was really a visionary. So this was what he wrote back in 2003. This is the future he envisioned, like the Human Genome Project being the foundation for a development of a better healthcare. So the idea was that getting knowledge of the genome and the function of its associated genes and the functional impact of all the mutations that could be mapped to the human population could help to develop a new way of conceiving medicine, shifting away from the classical approach and getting towards a more personalized medical approach. And in fact, he was right, and it only took about 10 to 15 years to get back to the goal of the 1,000 Genome Project. This is an article from 2014 that reports the achievement of this historical goal, the $1,000 genome, that was possible thanks to the development of a new sequencing platform, very, very different from the classical one, the Sanger method that you may have heard about. That was definitely a low throughput technique. Well, the next generation sequencers, the so-called next generation sequencers, allow the massive parallel sequencing of literally millions and millions of DNA fragments at the same time, providing an incredibly deep output that can be used to obtain, with a relatively low cost, the sequence of the genome of an entire individual. This is just an overview about how sequencing costs changed over time, and I'd like you to have a look at the scale on the y-axis because this is a logarithmic scale, okay? So this is the expected decrease of the cost in sequencing due to the Moore's law that actually forecasts a somewhat linear decrease in time. And something happened here between 2006 and 2010. And in this time frame, multiple independent companies developed new platforms for sequencing. that really allowed these massive parallel sequencing strategies to be finally available to the public. And as you can see, there was a massive drop of the sequencing cost per genome that, as you can see here, switched from the human genome from about $10 million to about 1,000 in a matter of 10 years or so. So we could say that today we can obtain the full sequence of a human genome of anybody for a very, very small cost that is now even lower than $1,000. It's even closer to maybe 100. So this is the next goal. And I'm pretty sure that this goal will be met within the next few years. So what to do with all this data? We can do plenty of things, but once again, I'd like you to really understand what's the power of these methodologies today. I just took a screenshot of the output of one of these sequencers that are really very expensive. So one of these things costs about 1 million euros, something like that. But once you have that, you can generate data with an incredible speed and incredible throughput. So keeping in mind, 13 years, $3 billion back in 2013, in 2003, sorry, what we get today in the matter of just a little bit less than 2 days of analysis for about $20,000 is that you get 6,000 gigabases. So it's an incredible amount of data. That's 6,000 billion base pairs. So the amount of material that would be sufficient to sequence not just a single genome, but 2,000 human genomes. And obviously this opens incredible opportunities to sequence a very large number of individuals and to collect all the information about the variants associated with different populations, for example, to understand which are the genetic basis of inter-individual phenotypic divergency. Okay? So why do some people develop some kind of illness when they become old and others do not? Why are some people more susceptible to infection by pathogens and others are more resistant? We've seen that, for example, with COVID very recently. You know, some people were more susceptible to a severe disease and had to be hospitalized, whereas others of the same age and apparently without any apparent, external feature, were doing pretty fine with infection. So understanding whether there is a genetic basis for this, for the development of diabetes, for the development of Alzheimer's disease and other stuff can be possible now thanks to these approaches. And so immediately after the end of the Human Genome Project, a number of other parallel projects started aimed at collecting genetic variability data. So we're talking here about the so-called HapMap project. Another one is the 1000 Genome Project. Well, the goal of these was to build a sort of a database of all the mutations that could be associated to virtually anybody. So this is obviously based on volunteers or on patients, so samples collected from universities, hospitals, and so on, being sequenced, put in a big database, and associated with phenotypic traits. By phenotypic traits, I mean presence of a disease, but also regular metrics like, age, weight, color of eyes, color of the skin, and so on. And we can try to understand, by analyzing in parallel these phenotypic trait data and genetic variation data, we can try to understand which are the associations. So which are the variants that drive a certain type of phenotypic trait. So the goal of these, of course, in the long run, would be to get to personalized medicine, as I told you, in terms of human genetics. Of course, then we can apply that potentially to many other organisms, but I'm trying to keep it the main focus of the talk at the moment on human. So we're moving progressively away through this first part of the genomic analysis in human, which was mainly focused on understanding the structure of the genome. Then there was a phase that is still a bit ongoing that concerns the understanding of the function of genes. So we have 20,000 protein-coding genes. We're not really sure about the exact function of all of them, but we're getting closer and closer to this goal. And the idea is that when we will have this information, we could be moving to this part here, which is improving the effectiveness of healthcare. So getting towards the so-called personalized medicine. The idea is that in the future, any people affected by a condition, a medical condition, will not be treated in a regular way like we do today. So everybody takes the very same kind of drug if you have a condition, but you could actually personalize the type of treatment based also on the genetic characteristics of the single individual. Of course, you need to have genetic profiling in order to do that. And at the moment, none of us has a complete genetic profile, but this is one of the goals that could be achieved in the near future, actually. It's one of the bold predictions of genomics by 2030, outlined by several members of the former Human Genome Project, including Eric Green. So a few points here are worth mentioning, for example. So the idea is that in the near future, by 2030, probably not, we will not get there by 2030, but maybe in 2040 we will, generating and analyzing the full sequence of a human genome would be a routine process, very, very easy from a laboratory point of view, as easy as purifying DNA, which, as most of you who work in a molecular biology laboratory know, it's a very, very easy, simple, and inexpensive process. Then there is the factor about the knowledge of the function of the genes. So at the moment, we still are unable to tell the function of all the 20,000 protein-coding genes of human, but we're getting progressively closer and closer to this goal. Another possible goal that could be met in a matter of a very few years would be to obtain genomic data in a manner as simple as collecting blood, for the regular blood test that everybody does a couple of times a year, right? And the final goal would be, of course, to develop new therapies that could potentially involve genetic modifications, the so-called gene therapy that people have been talking about for now at least a couple of decades. But once again, this knowledge allows us to move forward into this direction. And hopefully in the future we will be able to tackle some kind of diseases that at the moment are not curable with standard methodologies, with an intervention of the genetic mutation, which is the causative one. So what about the amount of genetic variation that we can see among individuals? This is a very important question. So how different are we from a genetic point of view? Of course, if we look at each other, we can immediately spot differences of our characteristics, but we may be overestimating the actual amount of genetic differences that underlies this morphological evident variability that we see. So if we would be able to sequence my genome and the genome of anybody of you, and we would compare the two sequences, we would spot, on average, about 4 million single nucleotide variations. This may seem quite a lot of mutations, but actually when you calculate which percentage of the full genome they do correspond to, it's far less than 1%. It's actually closer to 0.1%. So we could say that less than maybe a couple of nucleotides out of 1,000 are different between two random individuals from the same population. As you can see, there is a slightly higher amount of inter-individual variation in those populations from Africa and parts of Southern America that are, by definition, the ones that had the longest time to actually develop diversity. Because as you know, human life started in the Rift Valley, right? So this is where the oldest human populations of Homo sapiens actually were identifiable, okay? But is this level of variability high or low? How can we define it? Well, in order to define whether it is high or low, we need to maybe compare it with other species, what happens in other species. And, well, we can compare the phenotypes of different humans and clearly spot a Korean guy and say, okay, this is Korean, this is probably Nigerian, this is Slovenian. But what if we see a group of oysters or a group of mussels, the animals I'm working on? Well, you cannot really spot these differences. These guys look pretty much like each other, but maybe we are looking at the wrong traits. So we are biased, of course, we're humans. We are used to recognize our faces and to differentiate one people from the other due to many features. We're not really used at looking at the relevant traits of these animals. But truth to be told, the amount of genetic variation in these animals and more in general in invertebrates is higher by orders of magnitude compared to human. So once again, in human, we're talking about 0.1, 0.2% diversity between two random individuals. In these individuals, we're talking about 1 to 2% divergence between two random individuals. So 10 times higher. And this is, once again, another byproduct of our previous research carried out on a very few model organisms, mostly vertebrates, which are relatively simple in terms of genetics. And even when we talk about invertebrates, the usual models are Drosophila, which is a very simple organism. It's not a very good model to study biodiversity. So our model organisms are representative of probably less than 5% of the global biodiversity that we see in animals. So, another important thing, how much do we know about the impact of this interindividual variability? Well, still not as much as we would like. Because when we just look at the mutations that are associated with Mendelian inheritance, so the possibility of being transmitted from one generation to the others, these are the numbers. Basically, there are still about 1/3 of these inheritable diseases that have an uncertain genetic cause. And this is the gap that we need to fill in before really moving into precision personalized medicine. So how can we discover whether there is any uncharacterized mutation that is associated with a disease, for example, or with a trait of interest? Well, what is usually done is to sequence the genome of a group of individuals that share the very same feature we're interested into, so, for example, a disease, and to compare their associated genetic variations with those that are found in a control group which is not affected by that particular condition. These studies are called genome-wide association studies. So the outcome of that are these kind of graphs that are called Manhattan plot, simply because they do resemble somewhat the skyline of Manhattan with these skyscrapers. And the idea is that the skyscrapers correspond to regions in the genome that are statistically strongly associated with the trait of interest. Okay? To help you understand a bit better how the studies work, I'm going to move to a practical example, very recent one that deals with COVID. As I mentioned earlier, there are some patients that actually suffer from very severe infection, lung infection, as you know, and to the point of requiring hospitalization, whereas others in the same age range are doing particularly fine with infection. They basically do not seem to suffer that much. And there is a genetic explanation for that. The genetic explanation was actually provided by Svante Pääbo, a recent Nobel Prize winner, due to his studies about the origin of man, his work on Neanderthal in particular. And what Svante Pääbo and his collaboration did was to actually identify one of these skyscrapers here, located in chromosome 3, that was strongly associated with the patients that were hospitalized. Okay? So this is apparently a region of our genome that contains some mutations that are unfavorable in terms of outcome when somebody is infected by COVID-19. And the interesting thing is that this region here, that you can see here with the zoom, has been inherited from Neanderthal. And why was it inherited from Neanderthal? Well, because the Neanderthal man probably had a sort of a fitness advantage by having these particular variants, because it had a very strong response to viral infection in the past. We know that in the prehistoric times, the average lifespan was not very long. So it was very rare to see somebody survive more than 35 or 40 years, right? So basically the issue of people getting ill at 80 years old was not a problem, because everybody was already dead by 20, 30 years. Now, with increased lifespan, having such a strong response to viral infection can be a problem from elderly people. It's not a problem in the young people, but in the elderly people it is. So a trait that was originally favorable in the Neanderthal man to have a better response to viral infection, now in the modern man is a detrimental trait. And in fact, by calculating the quantitative effect of this kind of mutations, Svante Pääbo was able to demonstrate that anybody carrying this kind of Neanderthal haplotype had a 60% increased chance of ending up in a hospital after COVID-19 infection. So this is really quite relevant. In terms also of identifying people that should be prioritized in terms of early treatment and early diagnosis, right? So the point is, this increased availability of sequencing technologies are allowing us to move to a new level of analysis of inter-individual genetic variability. Usually we're always thinking about variability between people in terms of single mutations. A single nucleotide, which changes into another, maybe as an effect on the encoded amino acid. But this is actually just a tiny fraction of the variation that can be found within a species and among individuals. Actually, there is a lot of variation in this area here that we couldn't analyze until a few years ago. And these kind of variations are called structural variations. So very large region of the genome that can be absent in some individuals and present in others. There are chromosomal rearrangements, and these can have very, very important impact on the phenotype of an individual. Why do we know that? Well, because there are plenty of very large-scale genome sequencing projects that have very ambitious goals, such as obtaining all the sequences of all living vertebrates, like the Vertebrate Genome Project, or this other one, even more ambitious, the Heart Genome Project, that in perspective would like to collect the genomic information about all living organisms, all living eukaryotes. And by obtaining this data, we can actually move into comparative genomics and understand a bit better that there are features of the genome of these organisms that are quite different from the ones that we usually expect from a regular vertebrate, like human is. As a matter of fact, I've already told you that the human genome is not that special after all, when we compare it to most of other organisms. So now, for human and also for other organisms, we have the opportunity that we didn't have before to actually study this layer of variability among individuals. If we can obtain a single genome for 1,000 euros, well, we can invest money actually to collect more and more data and try to link this variability with disease, of course, but also with other stuff which is not as terrible as a disease. For example, to phenotypic traits that in other organisms are useful. For example, let's think about a plant, a crop, an apple, a tomato, a watermelon. We could try to understand which are the genetic variants that are associated with a better taste, a better color, for example, a higher resistance to drought, higher productivity, right? Why not? So let me start from the human, because as always, studies on human are the one that fuel research also in other domains. We have recently started to resequence the genomes of many humans using these large scale sequencing approaches. And we have started to see that there was quite a lot of sequence that was not shared between these resequenced individuals and the reference genome. Keep in mind that the reference genome was generated starting mostly from a single individual. So it's one genome. My genome is not equal to yours, obviously, and therefore, these resequencing data coming from several hundred individuals led to several million additional base pairs that were not present in the reference assembly. So the question now is, what is the function of all these accessory genomic regions that are not shared by all individuals? And then we need to develop a new concept, which is the concept of the pangenome, okay? So the point is that the single reference genome is not really that useful to represent the broader genetic diversity of the entire human population, because it's a single individual that has a particular genetic heritage. In this case, it was a mixed European and African individual, but that genome was not representative, for example, of the genetic variants that are most commonly found in Southeastern Asia, for example, or in native Northern Americans. So we are moving towards the new concept, the concept of the pangenome, which is the collection of all the genetic variants that are actually shared by all the individuals belonging to the same species. Well, the original definition has nothing to do with human, because it was originally formulated in a much more simple organism, in bacteria, okay? We know that bacteria can have accessory genes, for example, the ones that lead to antibiotic resistance. This is very, very relevant for that drug development. And obviously, these accessory genes can provide accessory phenotypic traits. For example, the ability to metabolize an antibiotic and survive whenever that antibiotic is provided to a patient, right? Well, the very same thing can also happen in other organisms, such as also human. So we're discriminating between the core elements of a genome, the ones which are shared by all individuals, and the dispensable elements of a genome, which are the ones that are only found in some individuals, but not shared by all of them. And we define an open pangenome whenever these accessory elements are very, very common and widespread, as it happens in bacteria. And we're talking about the closed pangenome, where these accessory elements are pretty rare, as it happens in human. Now the Human Pangenome Project is already very well developed. It was launched originally in 2019. It started collecting data from multiple individuals, and this is an outline of what the process of data collection and interpretation is. But you need to think that basically the goal is to try to link the function of these, the presence of these accessory genomic regions that are only found in some individuals but not in others, to a potential function. Just to provide a couple of examples about the first results that have been recently published, the first phase of the project was able to generate 47 genomes in total and to obtain 120 million accessory nucleotides that weren't present in the original genome. So this is quite a significant amount of data, reminding that the size of the human genome is about 3 billion nucleotides, okay? So we're actually adding here 5% of the sequence, pretty much, okay? And keeping in mind, in any case, that, as I told you, human is a species that, compared to others, has a very, very low level of inter-individual variability. So this makes you understand immediately that the functional relevance of these pangenomic architectures can be even bigger in other organisms. In human, still, they are quite relevant. I can bring you this example here about Chinese populations. In this case, the authors were able to implicate some of these accessory regions that are only present in people of Chinese descent, but not in Europeans, for example, to some traits that provide them with a better keratinization, a better response to ultraviolet radiation, a better performance in DNA repair. And this could be somehow maybe linked to the lower incidence of some kind of skin tumors, for example, in these populations compared to others. So we have somewhat of an explanation about the differences in incidence of some disease in different populations. Of course, there is also the contribution of environmental factors. Let's not forget about that. But sometimes there are important genetic components that shouldn't be forgotten. But pangenomic studies are definitely not limited to human. This is a very young and quickly expanding field of research that is now moving into other examples. This is a timeline. As you can see here, the animal pangenomes are only those that are circled in red here, so we have just a very few examples. And until 3-4 years ago, basically, we only had the example of the human pangenome. But just to getting towards the end of my presentation, I'd like to bring you a few examples of other pangenomes that target non-model species, just to make you understand what is their possible functional relevance. And I'm going to start with an example that is very close to me, because I was involved into this study, actually. And we did that for the common Mediterranean mussel that you all know as a seafood, probably. It may seem very, very simple as an organism, but actually its genome is extremely complex. It's a pangenome where about 1/3 of the genes are dispensable. So 1/3 of the genes are not shared by all individuals. They can be found in one population, but not in another one. What is the functional relevance of these dispensable genes? Well, this is a bit complex, but the take-home message is that their functions are clearly associated with immune response and survival. So we think that these genes that are only present in some individuals and not in others have been used along evolution to allow these species to adapt very well to some environmental niches. And this is the reason why now we see mussels widespread all over the planet in temperate waters, but we clearly see there is a genetic structure. So if we take the Mediterranean mussel, there is a clear pattern of gene presence substance variation that differentiate these mussels that are adapted, for example, to quite hot waters from the northern mussel, the Mytilus, which is only found in water with temperatures that are much, much lower in the winter, for example. And clearly, these genes that are missing in some populations and present in others can provide some accessory functions, such as a better tolerance of cold or a better tolerance of hot water. Mussel is just an example. There are just 3 or 4 slides more to show a couple of other nice examples that deal, in this case, with ecology. So this is a very, very small algae. This is a microscopic view. It's called Emiliana huxley. It's found everywhere. And the point is that there is a very large amount of genetic variation across these species. And the key questions that the authors of this paper wanted to ask was whether there was any genetic determinant that allowed these species to actually thrive to waters at the equator, at the poles. And, well, they did found that this species harbors a very complex pangenome with quite a lot of genes that can be missing in populations and present in others. And of course, these genes are involved into functions that are related with the ability to adapt to a given environmental niche. So once again, differences in temperature, differences in availability of light as well, since this is a photosynthetic organism, different availability of nutrients, and so on. There are also more practical implications, such as the pangenomes of pathogens. This is an example about what can happen in fungal pathogens, right? Well, there are some strains, as we know, of unicellular fungi that are pathogenic, others are not. And the determinants of pathogenicity actually are linked with dispensable genes. So this accessory fraction of the genome, which is really not shared by all strains. So we can have some strains that have high virulence and can lead to the death of the host, and others that are not that virulent and lead to the survival. Other practical implications deal, of course, with the development of sustainable crop production. This is a very important thing that we can do, And actually, pangenomic research is running very, very fast in the field of agronomy. Just to show you a couple of examples, one of the key questions that we can answer using these kind of approaches is, for example, why do oranges taste so good? Well, there is a clear explanation that comes from their genes, and more in particular, it is associated with the genes they carry and that are not shared with other type of plants very closely associated to them that live in the natural environment that are not very useful for us because they're not as tasty as oranges or as lemons, right? And this study was able to actually pinpoint which are the key genes that are involved into the synthesis of molecules that are responsible for the good taste of oranges. Similar study here about apples. So apples have very different shapes, very different colors, very different tastes, as we know. And it turns out that the major genetic determinants of these are linked with the fraction of the genome which is not shared by all of them. Okay? So the impact of gene presence substance variation is much more widespread than we think. And this is the final slide that I want to show you just to make you understand how complex the interplay between these accessory genomic locations can be. Sometimes species have to interact with pathogens. And pathogens over long evolutionary timeframes have clearly led to selection and to massive mortality events, right? So we know, for example, that a model species, which is Arabidopsis thaliana, this is the classical model species for plants, has different lineages, right? Different cultivars that are susceptible or not to infection by a specific type of bacteria, which is a Pseudomonas syringae. Different strains of Pseudomonas can have a detrimental effect on some strains, but not on others. And this depends on the combination between the presence of receptors of the host, so of the plants that can recognize the bacteria that carries or doesn't carry a specific antigen. Let's call it antigen, even though the definition is not correct in this case because, as you know, plants do not really have immunoglobulins. But the key idea is that only when there is a match between a specific molecular determinant carried by the pathogen and the receptor encoded by the genome of the plant, there can be a recognition and an efficient immune response that leads to the plant surviving fine. Whenever there is the wrong combination, so a pathogen that expresses a molecule which is not recognized by the receptor, simply because the receptor is not encoded by the genome of that particular Arabidopsis thaliana strain, then there is the development of the disease that can lead also to the death of the plant. Very similar things most likely happen also in animals. And we're just starting to understand how this complex interplay between host genomics and pathogen genomics work. And with this, I'd like to thank you all for being here. I'd be more than happy to answer any question you might have. And I'd also like to thank all my collaborations, both in Trieste, in Padova, and in Vigo in particular, that are currently working together with me on this fascinating topic, which is pangenomics. Specifically, we're doing that in muscle. Thank you very much.
Thank you very much, Marco. It was very interesting. It's also an ability to render easy things that are more complicated, no? And this is the story of pangenomics. I may start with the first question, which is also related to a big grant that we got a few years ago, a European grant. How do you believe that artificial intelligence, machine learning, these approaches can help us in this field of pangenomics?
So I think they could be very, very interesting in perspective, because as I mentioned, the association between the presence or absence of a gene, for example, that is dispensable and a given phenotypic trait is all done by statistical association, of course. The point is that there are literally thousands and thousands of variables when it comes to phenotypic variation. So what do we consider as phenotypic variation? There are some measurable traits that are very easy, discrete ones, like a plant that survives or dies after an infection. And there are some other traits that are actually quantitative and continuous. And these are very, very difficult to be analyzed. So at the moment, our study is mostly based on some traits that we are really interested into, like all the examples that I showed you were really focused on crop improvement. So the authors were really looking at the color of an apple, the presence or not of a surviving plant after an infection. But potentially, we might, if we manage to obtain a database of metadata associated with the single genomes, with all the phenotypic traits that could be potentially of interest, and at the same time we have a full genome sequence with presence-absence data for all the genes. Of course, these artificial intelligence approaches could help us to discover some hidden links, some interconnections that are really very distant from the ones we're looking for, right?
Oh, thanks, Marco. That was super interesting. And it seems pretty obvious to me the importance of what you presented to medicine. You mentioned at some point that in the future, full sequencing will be done in research laboratories routinely. Mm-hmm. And perhaps you've already alluded to the answer to my question I'm about to ask, but I would love to get your opinion on personalized medicine.
Yeah.
And whether you think whole genome sequencing will become part of routine clinical practice, or whether you see it more as a research tool to help identify more targeted approaches that could be more cost effective.
Yeah. This is a key question, of course. It has a lot of ethical implications. As you can imagine, with great power comes great responsibility, and this is one of the cases where we can really say that. I run very quickly through one slide that actually had a point that was very important, which was the structure of the human pangenome consortium, so the workflow. So let me get back to that, because I'd like to show you that whenever people try to put together such a big project, they need to consider something which is really central. So in this outline here, there's this part here which is really central and is connected with anything. And this is the embedded LC. LC means the ethical, legal, and social implications. This needs to be taken into account, and it's very hard for us as scientists to actually take into account these things, because we do research because we like it, of course. We are very curious people, and we are interested into understanding how life works, actually. But sometimes we do not really think about the implications that other people that are not really involved directly into scientific research could do. So the interest, the economic interest, of course, is always something that can be problematic. Just to better contextualize the problematic aspect of genome sequencing in terms of, for example, privacy, right? I didn't mention that the Genome Project, the Human Genome Project, was not a single one. There were two parallel genome projects, okay? The one I talked about was the one that was funded by mostly by the National Institute of Health of the United States. Of course, then there was the international collaboration, but it was basically publicly funded. In parallel, there was a privately funded project that initially had as a goal the obtainance of the human genome sequence that should have been patented, so covered, by public access. Luckily enough, there was an agreement that was signed quite early during the development of the project. It's called Bermuda Agreement, that actually established that all generated data from the Human Genome Project should have been immediately made public domain. Of course, imagine the potential issues also in terms of equitable access to healthcare, that having such information hidden behind paywall could have. So technology keeps running, will keep running. We will have the technologies to eventually obtain this genome sequence of any individual with a few heroes, okay? There's no way to avoid it, but there are ways, of course, to make sure that this doesn't result in inequities. But I don't think this is really the task for us as scientists. It should be the task of somebody else.
Hey, thanks for this comprehensive talk. I have a question or two, and first is maybe about the C-value paradox that you talked about at the beginning, and I feel it's kind of like then linking towards what you presented about the metazoan pangenome project in the end. Can you comment more about this C-value paradox? And for instance, is it, correlated with the evolutionary age and the ability to sustain also changes that are,
Okay, let me get back to that slide.
And stuff.
So the C-value paradox is something that immediately struck, the scientific community. So there were a lot of misconceptions initially about the expected complexity of the human genome. So initially, I told you that the scientists were expecting to see some 100,000 genes in the human genome based on their expectations from the previously available sequences of the genome of the yeast, right? So this is a unicellular fungi. Of course, we expect it to be a very simple organism with just a few thousand genes. So the point is that they were really surprised. Why they were really surprised? Because they were underestimating the aspect of the impact of alternative splicing from one side and the effect of epigenetic regulation on the other, right? So we have a lot of accessory sequences in our genome that are not protein coding. Actually, the exons, so the portion of the genes of the genome which are actually translated into protein are less than 2%. So what is the function of the other 98%? Well, in large part, in the past, it was considered to be junk DNA, so stuff that doesn't really serve any purpose. And it is in part true, of course, but there are plenty of regions that are extremely important to regulate the accessibility of the chromatin to proteins. These proteins interact with DNA and regulate very precisely when and at which level an mRNA needs to be produced. Let's think about a protein that has a function in digestion, okay? It's produced by pancreas. It needs to be produced only there. What would happen if that gene was expressed in the brain? Of course, our brain would be digested and none of us would be here, of course. So there's need to be a very strict regulation, not just in terms of tissue distribution, but also in terms of development. So this is the key. Something changes in a very dynamic way in terms of methylation profiles and allows to turn on and off specific genes with a very precise timing. So getting deeper into the C-value paradox, what really makes the difference here is, in part, the activity of transposable elements. So our genomes are actually invaded by elements that are extremely repetitive. So they are copied hundreds and thousands of times, okay? These are the result, most often, of very ancient viral infections. And as you know, viruses have the ability, in some cases, to be integrated in the genome and to actually copy and paste themselves multiple times. So some of these elements are not really viruses anymore, but they maintain the enzymatic activity. So they encode proteins that are, for example, the so-called integrases, transposases, also reverse transcriptases that could create new copies actively in the genome and lead to an expansion of this array of duplicated elements and to an expansion of the genome. So this is one of the aspects which, in fact, in a few organisms which have a very, very large size, such as the lungfish, which is a particular type of fish that is also able to survive living in the air during periods of droughts in a few rivers. Well, in this case, there is a a lack of control of the expansion of the elements that can actually continue expanding, expanding, and expanding. And in this case, we have genomes that are, as you can see, as big as 100 gigabases. So it's a very, very large size, more than 30 times larger than the human genome. Then, together with the C-value paradox, there is also maybe another more important paradox, which is the number of genes. This is something that we don't really consider usually. So if I take a genome of a sponge, for example, very simple organism, which is at the base of the tree of life in terms of animal evolution, it doesn't have differentiated tissues, of course. All the cells are pretty much the same. Well, the genome of the model organism for sponges, which is Amphimedon queenslandica, has something around 60,000 genes, 3 times more than human. Why is that? Well, because not all genes are involved into morphological complexity. There are plenty of genes that are involved into metabolic complexity. So the ability to metabolize many different compounds that are found in certain environments that are very different from the ones we are used to. So we are terrestrial species, a sponge is a marine one. We are not really exposed to very hard challenges during our life. Of course, there is some change in temperature, of course, but think about a marine species that lives close to the coast. Water level changes. These animals can be exposed to air. They can be exposed to sunlight directly. They can have a variability of salinity. For example, when it rains or when it doesn't rain, there could be shifts, incredible shifts in the amount of nutrients that are available. There could be some harmful algal blooms, so they can accumulate toxins, and they need to make sure not to die when they accumulate these toxins because they are filtrators. So all of these functions are needed in a given environment to allow survival of the species. So we don't need to ask ourselves why they look so simple from a morphological point of view. Most of the complexity of life forms is not really visible in terms of morphology, but is hidden in terms of metabolic activity.
Thank you for this interesting presentation. Actually, I was interested on, let's say, these evolutionary, a bit older animals, and maybe if we can take a look a bit closer on this variability, like how is it actually distributed in the chromosomes, and secondly, are there mostly like kind of structural variations or single gene variations, a bit more?
So this is a very complex question because different things happen in different organisms. I would say that generally speaking, vertebrates have a very low level of inter-individual variation in general, okay? And the more we go closer to mammals, the less important this variation is. So human is really not a very genetically diverse species, okay? So in general, higher diversity is linked with very, very large populations. Of course, human now has billions of individuals that are living at the same time, but this has been a very recent explosion. So when we look at the timeline of evolution, I mean, the past 200 years have been just a very, very tiny bit. Historically speaking, human population has been always very, very low compared to other species that are extremely widespread and very, very large. I'm talking in particular about invertebrates. Then there is another factor that contributes to increasing the genetic variability, which is the mode of reproduction, right? So we are used to a certain kind of reproduction, which is basically not linked to so-called spawning. When you think about spawning, so animals that release gametes in the water, for example, well, this is a mode of reproduction that is much more similar to what plants do, right? So in this case, it's water then that brings the sperm and the eggs together. In the case of plants, of course, it's either wind or pollinator insects. But the idea is that it's a much more promiscuous way of reproducing, right? So you have a production of potentially millions and millions of offspring And it's a different evolutionary strategy of parental investment. So in these animals, you don't have parental investment at all. You just produce an extremely large amount of offspring, knowing that a very tiny fraction of them will survive, right? On the other hand, we have parental investment. So it's not just a matter of taxonomy. It's more a matter of the way these animals reproduce. So for example, let's talk about two very closely related examples from a taxonomical point of view. Well, if I take an octopus and a mussel, they're both mollusks, so they're evolutionarily related with each other, but they have a completely different mode of reproduction. The octopus has a strong parental investment, and the pangenome is closed. So there is almost no variation in terms of dispensable genes. Mussels, oysters, clams, scallops have open pangenomes as a result of the very large population size. In ecology, we call that effective population size and mode of reproduction. And to quickly answer about the second part of the question, how these regions are distributed, well, I mostly study that at the gene level. So I'm looking specifically for variants that are at least as big as a full gene, protein coding gene, okay? And they are distributed pretty much equally, okay? So we're talking about kilobase scale indels, sometimes megabase scale, okay? And so far it looks like there are some hotspots of hypervariability, but in general they are distributed everywhere. So it's not something that only affects a single chromosome, for example, but it's more widespread, I would say. We're still trying to find a precise answer for that. There's no precise rule because, as I mentioned, some species display a more uniform distribution of these dispensable regions. Others have clear hotspots associated maybe with a single arm of a chromosome.
Other questions? We have 5 minutes more for another one.
Hello. So I want to clarify this. So the human genome was sequenced by a single person.
So let me get back to the slide. Basically, most of the reference genome was obtained from a single individual. The point is that this was a very large collaborative effort. So multiple laboratories were sequencing at the same time, and there were some gaps to be filled. And of course, they didn't extract kilograms of DNA from that single person. They had backup samples. So it turned out that by reanalyzing the finalized version of the genome, the reference genome was by 70% most likely obtained from a single individual. Okay, I think this is the slide I was talking about. So basically, if you look at this graph here, this is the individual contribution of different individuals to the reference genome. This is the main individual. So about 65% of the current reference genome derives from a single individual. And this individual is expected to be of mixed European and African descent. And a very tiny part of this individual had a distant descent from Eastern Asia. So maybe one of the grandparents was Eastern Asian. And all these other parts derive from other individuals. So at least this large part here of unknown means that there are probably several dozen peoples that contributed to the assembly. So it's a chimeric assembly, basically. It's not the genome of a single person, but it's mostly from a single person with several gaps that have been filled with genomic data from other individuals. This is because, of course, the old-style Sanger sequencing required a very high amount of DNA, and it was impossible to actually ship sufficient amount of DNA from the very same individual to all the dozens of laboratories that were sequencing at the same time in different parts of the world. So somebody had to use backup material, and back then we didn't really realize what was the impact of inter-individual genetic variation, and we didn't think that this would have affected quite a lot the sequence of the human genome itself. But now we know that this is somewhat of an issue, and in fact, last year we had the release of an updated version of the human genome, which is now obtained from a single individual, and is also more complete. So we were able finally to finalize, officially finalize, and close the last gap of the human genome only nearly 40 years after the start of the project in 1990.
Good, thank you. Just a short one.
Yeah, sure.
So what's the precision of this sequencing? So what's the
Okay,
error?
the accuracy of current methods is about 99.99%. Okay, so the chances of having an error are very, very tiny, and basically there have been two main points that have been addressed in terms of development of novel sequencing approaches. One is accuracy, so reducing error rate, and the other one is increasing the size of the fraction of the DNA that could be read. So Sanger sequencing originally was very accurate. It had a very low error rate, but the so-called reads, so the fragments that could be determined, were very short, just a few hundred nucleotides. Then we moved towards next generation sequencing. The new approach, very short reads, 100 base pairs, but very, very accurate, and with a very low cost. So it's a trade-off between length and accuracy. And now we are finally back to very long reads. We're talking about kilobases of, so several thousand nucleotides with very high accuracy, with the new PacBio methods, for example.
Just a small comment about reference genome. Actually, just now, it started the new European project, which is called Genome of Europe, who will be, let's say, the new reference pan-genome for all Europe, and also Slovenia and Italy and are taking part of it.
It makes sense. So, another comment which is important, as maybe this other part of the graph here, is that basically, for obvious reasons linked to the imbalance between science, European, North America, and all the other parts of the world, the overwhelming majority of the genetic data that are currently deposited into polymorphism databases are from European people. So definitely we need to improve the representation of other ethnic groups, because this could be the only way to make to really attain an equal accessibility to improved healthcare to everybody.
So,
maybe Shed.
I mean, you can ask the last question and then we move downstairs.
Thank you for this amazing vision of your research and what is still to be done. I'm a little bit totally actually out of this topic, but I say, okay, I will come and I will learn something, and I did, and thank you very much. My question, very simple, can you say something about the technology of genome sequencing?
Okay.
Thank you.
So, well, I usually explain the novelties of genome sequencing to my students in about 6 hours of lesson, so I'm trying to condense it to 30 seconds. Things that change quite a lot are linked with the ability to process in parallel millions of fragments of DNA at the same time. Whereas in the past, the old style Sanger sequencing was focused on a single molecule at a time. So now we have ways to actually compartmentalize millions of reactions on millions of individual DNA molecules that can be sequenced by imaging, let's say. So, yeah, imaging means that, well, okay, you have like a sort of a very tiny plate which looks like the cover of a microscope, okay? This is the size of the unit that's being analyzed. You actually immobilize DNA molecules over this slide, and this slide is actually placed under an optical reader. And there are millions and millions of spots that are colored sequentially in different manner with 4 colors. Each color corresponds to a given nucleotide, A, T, G, and C. And this optical reader takes a photograph each number of seconds, and it does sequentially 100 times, and the sequence of colors determines the precise sequence that was obtained at a single spot. So you get millions of spots, and this is the way that the sequence is acquired. The chemistry behind that is very complex, because we have a polymerase, a DNA polymerase that adds 1 nucleotide at a time. The nucleotide is connected, is linked with fluorophore. So basically you have the addition of a single base at a time connected with 1 out of 4 fluorophore. Each fluorophore has a different color, of course, depending on the type of nucleotide that is added. And at each step, there is this high-resolution photograph which is taken, and the photograph is converted into a letter. And then you do that 100 times, and you get 1 million of sequences that derive from this color to letter conversion, basically. I hope I have explained you. Yeah.
Thank you very much, Marco. We are out of time. Thank you all of you for the questions, for the presence here. Thank you, Andrea, for the organization of the event. And we can move downstairs now.